Good evening, and thank you for joining us. Without further delay, I'm going to introduce today's keynote speaker, Professor Sasselov. We have the pleasure of welcoming a great expert, astrophysics and the formation of planets. Professor Sasselov was born in Bulgaria, and he is professor from the University of Sofia, and he's got his PhD in Toronto. Got to Harvard back in 1990 as a postdoc stay, and he is full professor since 1998. Phillips Professor of Astronomy, Director of the Origins of Life Initiative, Senior Advisor in the Sciences for Advanced Study. Origins of Life Initiative joins biologists, chemists, and astronomers in search for the origins of life. To answer a very interesting question, is it common, life in the universe? And to place this into a context and to understand the way the formation of planets has evolved, I'd like to mention that 25 years ago, we only knew our solar system, one planetary system, and now we have 3,600 exoplanets been confirmed. Professor Sasselov has contributed in a significant mode to this thanks to the analysis of data stemming from Kepler mission from NASA. I'd like to say that Kepler's mission has confirmed more than 2,300 planets using the methods of the transits, planetary transit. Thanks to this initiative, Professor Sasselov is trying to understand the origin of life in our planet, and he might understand the way it's stemming in other Earth-like planets. And he's going to talk about the bridge, establishing astrophysics and the connection with the biology. Thank you very much. You have the floor, Professor, and welcome to our meeting.
Good evening, everybody. Thank you, Nuria, for the nice introduction. I want to thank you all for coming tonight. Of course, the opportunity for me to be here, the thanks go to the foundation, BBVA. I'm very happy to be here. Everybody is very kind and very hospitable. Thank you very much. As you can see from my title, I want to talk to you tonight on the topic of an age-old question about life in the universe. And this question for my talk tonight will be covered by two separate questions. One, as you can see from the title of my talk, is the question about is there life on planets beyond planet Earth? The second question is more basic. What is the nature and the origin of the phenomenon that we call life? In this second question, I will elect tonight not to talk about the historical context. Instead, I'm asking the question rather as a question of general mechanism and essential attributes. So in that sense, I'm going to really try to talk about life despite the fact that the scientific community still does not agree on a definition of what life is. As a result of that, I will try to convince you that it is still possible to search for life beyond Earth and different types of life, while we are still trying to understand the nature of that phenomenon. So let's begin. What you're going to hear tonight is, of course, my personal perspective on this very exciting topic. I'm an astronomer, so it's the astronomer's perspective. And we astronomers like to look at the night sky a lot, like I know everybody does. So I would like to start with some of the basics relating to the first question, which is, where do we search for life? So what has happened in the last several decades, we astronomers managed to complete a very good census of our galaxy. We know how many stars are in the galaxy. We know what kind of stars they are. And the basic answer from that census is that the galaxy has a lot of stars. And while this number is very large, you have to remember that the galaxy is also a very, very large place. So the distances between the stars are very large. And although we like to think of the stars as being pretty on pictures like this, actually they're very tiny and very far from each other. Thanks to our new technologies and telescopes like the Hubble Space Telescope, we have also managed to understand, in the past 30 years mostly, quite a bit about stars and their development, as we would say, the life of stars. We know how stars form. They form by essentially condensing under their own weight from the hydrogen gas that is in between the stars in the galaxy, in places like the Eagle Nebula here, in places like the nebula in Orion. Stars form, and we see them, how they actually do that in such places. What happens next when a star is condensing out of the cloud is you see the formation of a disk circling around the newly formed star. If you look in this particular picture of the Orion Nebula, which is right in the middle of the constellation of Orion, you will see young stars surrounded by different disks. And in these inserts taken with the Hubble Space Telescope, you can see that some disks are face on and others are edge on. So you can see that. These disks are very important to the story I'm telling you tonight, because this is the place where the dust particles and some of the gas in the disk is concentrated to a point at which planets begin to form. And planets usually form when there is enough concentration of dust particles, what we call rock, when it's actually already condensed to become a planet. So the places where planets form are those disks, which in higher resolution with our new facilities, like the ALMA radio telescope, can be seen in finer detail. What you see here is an image of one of those disks, nearby disks, in which the star is right in the middle. It's very small. You cannot see it because the light in which this image is taken is really more into the radio waves. And so the star is fairly faint there. So instead, you can see the disk that surrounds the star. And as you can see, the disk already has some structure, which many of my colleagues interpret as planets starting to forming in this disk. So this is where the story begins, with the formation of the planets. And these are the places where there is enough concentration of the elements that are required for what happens next in planets like the Earth, which is the emergence of a complex chemical system, which we eventually end up calling life. And so planets are places, the places where we would be looking for in our search for life beyond Earth. So there are two options in our search. First option is the obvious. That is the option in which we look for life on other planets in our own solar system. Here you see the planet Saturn, which has the beautiful rings, which in certain sense resemble the protoplanetary disk that I showed you in the previous image, although they're very different, and that's why we call it a ring, the rings of Saturn, as opposed to disks of Saturn. So Saturn is one of those planets in the solar system, but it's not really a planet which is interesting from the point of view of searching for life. You would ask, why not? Well, it's a gas giant. Most of the constitution of planet Saturn is made of hydrogen and helium. And from a chemist's point of view, that is considered a very dilute environment. Not much chemistry can occur, and especially chemistry of big molecules like the ones which we're interested in doing. However, there are other places in the solar system where it is worthwhile to look for life. Of course, all of us know about Mars and how Mars is very Earth-like compared to all the other planets in the solar system, although it's quite cold and inhospitable today. In the past, Mars, as we now know from the geology of Mars, must have resembled very much early Earth. Warmer, with more dense atmosphere, and with water, liquid water on the surface. But there are other places in the solar system, and not surprisingly, I'm showing you Saturn, not only because it's a beautiful picture, but because Saturn has a moon, which is called Enceladus, and which is responsible for that most distant ring of the Saturn rings in this picture. If you're looking at Saturn with a small telescope at home, you will only see the bright inner parts of the rings. This is what we usually see on pictures. This is a picture taken from Saturn by the Cassini spacecraft, and it can see much more than what we can see from the Earth easily. So this ring, which is called the E-ring, the most outside one, is very thin, but it's produced by water which comes out of geysers, literal geysers, from one of the moons. The moon, actually, Enceladus, orbits exactly where this ring is. You can't see it on that picture, but it's there, and it produces that remnant material. Enceladus and another moon of Jupiter are of great interest to people looking for life beyond Earth in the solar system, because we know now that both of these moons have liquid water oceans under a crust of ice. Of course, they're covered with a crust of ice because Jupiter and Saturn are so far away from the sun that the ambient temperature there is very low, so any water would immediately freeze. The moons are small, they're not big planets like the Earth, so they don't have atmospheres. And so all you have is a solid crust of ice, but under that solid crust of ice, you have a liquid ocean. And in the case of Enceladus, there is enough energy from the tidal interactions of the different moons that that ocean is being kept relatively warm. The water is obviously liquid. And in addition, the crust is being stretched all the time, so cracks form in the ice crust, and those geysers spew out water. I don't know whether we will be successful in finding life in either of those places, Mars, Enceladus, or Europa. I do support, however, that we go and explore, and I hope that in the next 10 years we'll actually have real data telling us whether there is any evidence for life on either of them. However, for me personally, there was a big transition and a breakthrough that occurred in the last 5 years, even as early as 10 years ago, when I decided that I will put most of my effort in searching for life, not in the solar system, but on planets orbiting other stars, or as we call them, exoplanets. And the reason for that is quite practical. I want to be successful in that search, and I estimate the chances of discovering life other than Earth life in the solar system are very low. Not zero, but low. While at the same time, just in the last 2 years, we learned that planets like the Earth around other stars are very, very common, much more common than we expected before. So I'm putting my bets on the possibility that because I have a big statistical sample, many, many possibilities to explore, I'm more likely to succeed in searching for life on exoplanets. So let's see what is the situation with exoplanets. Well, as I told you, it's only in the last 2 or 3 years that we finally have the sensors for those as well. And now we know that there are more planets in the galaxy than there are stars. Of course, this is any kind of planets, including planets like Saturn. So what is exactly the kind of planets that we are looking for? Well, this realization came from the scientific analysis of the data produced by the NASA Kepler mission. And as Nuria already mentioned, this is the reason why I'm actually here tonight, because that is the underlying motivation for the search for life on those planets. The Kepler mission consists of a telescope, a modest-sized telescope, but dedicated to pointing in one particular direction on the sky in the constellation of Cygnus and Lyra, where for a duration of 4 years, it observed without any interruption all the stars in that field. The reason we planned the Kepler mission in that particular way, observing only the same stars for 4 years, was because at the time when we, in fact, proposed the mission and designed it, which was in 1999, 2000, we didn't know whether there are many planets around other stars. That number was not known. So in a sense, what we were trying to accomplish is simply a statistical estimate, a census again, which is useful for knowing how often planets like the Earth occur in the universe or around other stars in our galaxy. So that was the design of that mission. The technique which was used to discover the planets is somewhat unusual, but it is important to my topic today. So I want you to see the little insert which is in the upper right corner of this image. The technique is called the transiting method technique. It relies on planetary systems, on planets, which are aligned with respect to us and the telescope so that the planet passes in front of its star, literally in front of its star. So the orbit of the planet is in the same plane as our observing position here on Earth, or in the solar system in general. So a little eclipse occurs, and that eclipse diminishes the light of the star a bit, and occurs periodically. Every time the planet comes back, there is another eclipse. Those eclipses are called transits because they don't block the star completely, they just block a little bit of the light from the star. Obviously, we are not going to discover many planets that way because the orientation has to be just right. So the rough estimate is that about 1 to 2% of all such planets will have the fortunate orientation to show transits. But if we observe 1 million stars and there are sufficiently large number of planets, this will occur multiple times for multiple cases. And so this is the goal of the proposed mission. It worked beautifully. In fact, it worked better than we expected, partly because there are more planets than we expected when we wrote the proposal. So Kepler did its work, and that work and the whole mission was officially completed this summer. So these are the results from what was presented this summer, and the papers written and already submitted. So as you can see, this is as of June of this summer. So this is the total number of planet candidates, and by that is mean that confirmed planets, the total number is 4,000 and a little bit more. The different colors is simply to say these are the previously measured ones by Kepler, and the yellow color were just the final additional ones which were reported this summer in June. But as you can see, we have planets of all different types, from Jupiters to Neptunes, of course, to many Earths, and actually planets smaller in size than the Earth. The smallest ones we've detected is actually even smaller than Mercury. Of course, the smallest planets in the lower left corner, where also the orbital period is extremely short. So they transit much more often, and that increases the possibility for us to discover them and detect them. The planets that we are really interested in detecting are in the lower right corner of this diagram, and as you can see, that corner is relatively empty. And this is because it's so much more difficult to detect a planet which only transits 2 or 3 times during the whole 4-year mission of Kepler. However, we detected sufficiently large number, and the overall statistical conclusion of that is that there are a lot of small planets, and particularly planets of sizes comparable to Earth. In this diagram of planets per star with these periods and planet radius, where the gray and blue areas is where the big planets, the giant planets like Jupiter and Saturn. And as you can see, the number starts going up. These are the bars and the dark points, which show how the numbers go up as you go to smaller planets, although it's a lot more difficult to detect the small planets. So we are missing quite a few here, while we can see all of these. So now we know that Earth and the slightly larger versions of Earth, which we call super-Earths, are the most common in the census of the galaxy when it comes to planets, which is very good news. It also is important to realize that those planets which are 30 to 80% larger than the Earth in size, and we call them super-Earths for that reason, are a very interesting member of the family of Earth-like planets, because they're just slightly bigger than the Earth, but for all other purposes, they're just similar to Earth in terms of what their surface conditions would be. In certain sense, they're even better than the Earth. Being slightly bigger, it's easier for them to retain an atmosphere and to be more stable in many other ways. So if you think about climate similar to Earth, the planet being a little bit bigger actually helps that climate being more stable than if the planet is smaller. Very good example for that in our own solar system is Mars. Mars was very similar to Earth in its conditions in the beginning, but over the last 4 billion years, because it's so much smaller, it lost essentially most of its atmosphere. And that's not very good if you live on the surface. It's not good for chemistry, it's not good for life in general. So super-Earths have to be considered as part of this family. And you may ask, well, how come it's a new type of planet? That's because in our own solar system, there is no such planet. And that's just how it happens. Planet formation is a somewhat random process, and you get a certain number of planets, and whatever the dice rolled, you get those planets. So, in other words, you can say that the largest super-Earth in our solar system is the Earth itself, because Earth is part of this family. So, back to our main question. How do we find the census of those planets that we really care about, where we want to search for life? The ones that are really like the Earth in many ways, not just in size. And then you have to invoke and know the distance of that planet from its star. Fortunately, with Kepler, you do that. With Kepler, because of the repeated transits, you know the period, and knowing the star, you know the distance of the orbit. In fact, transiting planets are very good for many reasons. The transit method is not only good for discovering planets and getting their period and the distance of the orbit, but as you can tell, because it's a miniature eclipse, the amount of dimming that you get will also give you the radius of the planet. So you end up also with the size of the planet. As you noticed, Kepler immediately produces sizes of all its planets. And that's a very important and good thing, because when you match the size and the mass of the planet, you get its mean density, and you can say something about its composition. Is it a rocky planet, or is it a gas planet, like Saturn? Anyway, so the bottom line here is that we have more information, and that additional information is very important. As you can see again from the report, final report from the Kepler team, again as of June this summer, we now have a sizable number of planets which are in the so-called habitable zone of their stars. The habitable zone here is marked with the green, green areas on the diagram, where what is being compared is the surface temperature of the star on one hand, because our planets have different host stars. Not all of them are exactly like the Sun, which is the case up above near the top, where you see just barely see Venus, Earth, and Mars. That's where the Sun is in temperature. You see how they're lined up at the same temperature? That's because all three planets, Venus, Earth, and Mars, of course, orbit the same star, which is our Sun. So that's the star for our planets. And then all the other ones, as you can see, there are a few that are hotter, just barely hotter, but the majority of them orbit stars that are less hot and actually smaller. The sizes that are shown there reflect the actual size difference between the host stars. And then what is compared to that is the energy received by the planet. In other words, the heat that you get from the star. And being at different orbits will allow you to receive a different amount of heat. If you're too close, let's say 3 times the energy received by the star compared to what the Earth gets, which is at 1, you get too hot. The climate is not very good for any chemistry. It's just hotter than Venus. Venus is extremely hot on the surface. If you're too far, then you're too cold. And then the best solvent for life as we know it, which is water, gets completely frozen. And a lot of the chemistry cannot proceed. So this is what the habitable zone is, and this is how we use it. Another way to present it is in a more graphical way, in which the green area again is given as a distance from the star. And so here in this illustration, you can see that our inner solar system compares favorably to the TRAPPIST-1 system for 3 of its planets. But you have to remember that the TRAPPIST-1 star is so much smaller than our Sun, actually 10 times smaller, that this upper part had to be enlarged 25 times in order for you to see it. Otherwise, it is really all fitting within that very small area in the middle of our solar system to scale. So the question then, now that Kepler is finished, is what is that fraction? And the answer is somewhere between 5 and 15%. And that is a very, very important number. And you will say, well, it's not very accurate anyway, 5 to 15%. Couldn't you do something better with $500 million, which is the price for Kepler? Actually, it's a wonderfully good number compared to what we knew before Kepler, which was nothing. We didn't do anything. It could be 0, it could be, 1,000 times more than this. So now that we know this number, we can do a very important estimate for the purpose which I'm excited about, looking for life. And that estimate is to say, given that number, if I look at the nearest stars, the ones that I can really study, stars and planets which I can really study with the technology we have today and with the telescopes and instruments that either we have today or we are building, which are already being built and we know how to put them together. How many such planets will be in the nearest neighborhood? And let's define the nearest neighborhood as 30 light years, which is really very close compared to the size of the galaxy. Well, there is the estimate. Within 30 light years, there should be about 60 planets potentially like Earth. 60. Before Kepler, the people who were optimistic about this number would give the number as 1 or 0. So now after Kepler, the number is 60. That changes the game completely. Before Kepler, we didn't have the technology to try this experiment, to look for life on Earth-like planets, because the nearest Earth-like planets would have been too far away. Now that we know the number, we know all of these 392 stars. You see the number there? Within 30 light years, we have 392 stars. Why is the number 392? Because this is exactly the number of stars they are. We know all of them, literally. We haven't missed a single one. All the stars are well accounted for because this is so close to us. And with Kepler's fraction of 5 to 15%, the rough number is about 60 planets. Most of them, not surprisingly, come from this big red column in the histogram. The big red column in the histogram is the most common star in the galaxy, the most common star in the universe. These are the little red dwarfs. We call them M dwarfs, but they're really small red stars. For every sun there in the galaxy, there are at least 12 of those small ones. TRAPPIST-1 is one, Proxima Centauri is another one. There are many of them. You don't see them when you sit and look at the sky at night because they're also very dim. They're the closest, but they're also not very bright. However, they have planets and they have a habitable zone. So there are plenty of planets in the habitable zone of those small stars that potentially could be good targets for us. And we already mentioned two, or I mentioned two, TRAPPIST-1 and Proxima b. So now that we know where to look, which is in those potentially habitable planets, what do we do about it? Well, first we have to discover them, but that already has been taken care of by NASA funding another project, which is follow-up on Kepler, and it's called TESS. It is a project which is smaller than Kepler, cheaper than Kepler, because all it's doing is discovering those nearest transiting planets, nearest to us. So it's doing a whole sky survey as opposed to staring just in one direction as Kepler did. So that spacecraft is already built. It's now being finally tested and is scheduled for launch in the end of March next spring, so in 6 months. It will start its survey. It will only take about 3 to 4 years to complete the survey, and then we'll have all these targets that we need. So now that we know the targets, what do we do next? That's the question. Well, things starting to get more difficult from now on. The first thing is, how do we search for life on those distant planets? It turns out that, though difficult, this is something which we know how to do. There is a good example here of a very common bacterium on planet Earth, and very important to the geochemistry of the whole planet. They're called cyanobacteria. They're green, and essentially the green stuff, which is life, produces the silver stuff, which is oxygen gas. So there is one example in which life on a planet, planet Earth, literally transformed the planet. It transformed the atmosphere and the surface of the planet over a period of its history of about 3 to 4 billion years. What happened, thanks to mostly cyanobacteria, is from the very beginning of the active biosphere on the Earth, over the first 1.5 billion years, that oxygen eventually came to dominate the gases in the atmosphere and the water in the oceans. So today we live on a planet which is very rich in oxygen gas. But that is not normal if the planet doesn't have life. A planet that does not have life should not have free oxygen in water or in the atmosphere, because it's very reactive, chemically reactive, and very quickly is locked up into oxides and other ways chemically. So no other planet in the solar system has free oxygen in its atmosphere. Earth is the only one. So we call this a biosignature. That is, it is an indicator, if you see it on another planet, that something really interesting is going on its surface. And it's not pure geochemistry. So this is what we are looking for. And how do we look for oxygen gas? We do it by remote sensing. Remote sensing is simply a technique which we have been using now for 50 years, since the beginning of the space age, and its use increases and improves with time. It is how we usually look at the Earth from orbit and measure different gases and different surface features. That is called remote sensing. Most of this is done with relatively small devices, which are called spectrometers, which take the light, usually visible light, blue light, all the way to infrared light, and split it into its colors. We call this a spectrum, hence the instrument is called a spectrometer. So when you split light into its colors, different molecules like oxygen gas or methane or some surface feature, they have different fingerprints or signatures, clues in that color at different, as we say, wavelengths. Different parts of the color reflect different molecules. The signatures are very, very precise. You cannot really mistake one for the other. One way to say it, there is a lot of information in the color spectrum of light. So it can allow you to be very precise and measure different gases. It can also allow you to measure very small concentration of gases, because even a very small concentration of gas shows up in a spectrometer. Remote sensing is what we do also on the ground. There is one example from the Soccer World Club in Germany. So what the spectrometer, which is shown here in the left side, this metal thing does, it scans the audience, the people who are in the audience, for vapors of alcohol. And guess what happened here? There is one very hot pixel there. That means that there is some German fan there that had too many beers. And this is the breath of that person. It can be seen by the little spectrometer here. And of course, I'm sure some people were sent to visit him, I assume, and tell him to not drink that much. So this is called remote sensing. We can do this. We know how to do it. And in this case, what we're doing is we put this same kind of spectrometers at the back end of a big telescope, and we look at the stars, and in this case, at the planets that are orbiting those other stars. And we already started the process when we realized how powerful this is. In our particular team, we spent particular energy and work to build one of those, but much bigger and we think much better, to study stars and planets that way. It's called, for lack of better name, Harps North, and it's here in Spain. And when I say here, it's actually on the Canary Islands, but it is in Spain, and it's a collaboration between Harvard and Switzerland and Spain and Italy. And in one of my few trips up to the mountain, I stood next to the big vacuum tank. We keep it in vacuum so that it's stable. That's a requirement. And in fact, the entire room in which this vacuum tank is closed and hasn't been opened for the last 5 years because we keep the environment very stable. It's on the beautiful mountain peak in La Palma. So that is what we would say a prototype which we now are using to build an even bigger instrument than this that I will mention in a moment. So what is it that we do with it? There are 3 basic ways in which we can do this remote sensing and detect the signatures of life, like oxygen gas on a distant planet. And I want to show you the 3 of them here. Let's start with the easiest to understand one, which is at the bottom. The bottom 2 panels show you what we call direct imaging. And it's exactly what the word means, direct imaging. If we could see the planets directly with the telescope, like bright spots, you see the star there is blocked. The light from the star is by far the strongest, but we have telescopes with which we can block the light of the star, and then we see just the glow, the reflected light from those planets. Then we can train the spectrometer, the ones that you just saw, to one of them, say planet C, and get a trace of the colors in the light. As I said, we call this a spectrum. But these are the fingerprints which tell you if there is methane there, carbon dioxide, oxygen gas. So direct imaging is kind of the best way, if you will, to do this, but it's the most difficult because we only have a handful of planets for which we can do this, and they are not Earth-like at all. They're bigger than Jupiter. So we cannot use direct imaging for now. That is something which will come in the future. Then we have the top two panels. One of them is called transmission spectroscopy. And again, it is spectroscopy which uses transmitted light. What does this mean? This only works for transiting planets. Remember, we talked about transiting planets and the transiting method. These are planets which have been discovered to transit in front of their star. So occasionally, every orbit, they spend a few hours in front of the star. Again, we don't see with the telescope the image of the star and the dark spot of the planet, but we see the integrated light. And that integrated light will contain light that is passing through the atmosphere of the planet. Transmission, that's why the word transmission. The light from the star is passing through the atmosphere, the annulus, the atmosphere of the planet, and is coming to us, to our telescope. If we are good enough of doing that and have a precise instrument, we will see signatures of those gases that imprinted when the light passed through the atmosphere. The same way we do it here on Earth, but now on a cosmic scale. That is called transmission spectroscopy. And then, of course, there is the emission spectroscopy, where there are no transits, which is for many planets. 98% of the planets will not have transits. And there you have to count on the different phases in the emitted light changing with phase. That's also difficult. It turns out that of all those three methods of accomplishing the detection, the most mature, the one for which we have the technology and which we know will be successful if we have those planets to work with, is the transmission spectroscopy method. And so our goal is to use transmission spectroscopy to detect oxygen gas in Earth-like planets around the nearby stars. So again, the light is passing through the atmosphere of the planet, and if there is oxygen gas, it will show up in the spectrum. So this is kind of a way to show you how this works. Although we see none of the planet, we see only a bright spot where the star is, but we know when the transit is. And while the planet is transiting, the spectroscopy of the combined light from the star and the planet passing light through its atmosphere will have a signature at a very specific part in the spectrum, at colors around yellow and red to our human eye, or wavelengths between 760 and 770 nanometers, if you like that, which is due to oxygen gas. And this is the exact shape of that signature. Molecules have many ways in which they vibrate and rotate, and that's why their spectral signatures, that is, their fingerprints, are very specific. As you can see, there is a lot of information in this little dark spot in the spectrum, from which we can tell precisely that this is oxygen gas and nothing else. And also, because the planet is moving through while it's passing in front of its star, even if there is the same gas in the atmosphere of the planet that we're observing from, which there is, it's Earth. Remember, our telescope is at the bottom of the atmosphere. So there is oxygen in our own atmosphere, which will also show the same signature. But because the transiting planet is moving, there is an effect which was discovered 200 years ago by Doppler, called the Doppler shift. And that Doppler shift will allow the two set of lines to separate from each other. and we can see that. The project is pioneered and led actually by a Spanish astronomer, Mercedes Lopez Morales, who is working very hard on this right now. And the funny characters there with the GMT plus G-clef is simply what is on the next slide. This big telescope, which is being built right now, is called the Giant Magellan Telescope, GMT. And the instrument that is supposed to do this measurement is called, again, somewhat unimaginatively, G-clef. So this instrument is a bigger version of the one which is currently on La Palma that I showed you, Harps North. And as I said, it's being built. It's very precise, very high resolution. The most important thing is what is here at the bottom line, which says first light instrument. In astronomy, we build these very large telescopes as a community. It's not a single department or even a single country. The Giant Magellan Telescope is a consortium between a dozen universities and institutes in the United States and several countries, Australia, South Korea, and so on. So time using that telescope is very precious because it's an expensive instrument and everybody wants to use it. By the fact that the community decided to have this instrument, this spectrometer, as the first light instrument when there will be no other instrument on this big telescope, tells you that everybody wants to use it for things like the one which I just told you that Mercedes Lopez Morales is leading, which is detecting that oxygen gas on another Earth-like planet. So, as you can see, we have a very detailed plan how to do this. It's already funded. The instruments are being built. So it is not a fantasy of the future. It's actually going to happen. And whether it's going to happen in 3 years from now or 5 years from now, that's just a matter of when we're going to be ready and do those measurements. But in astronomy, just like in physics, you have to plan these things well in advance, and the planning has already been done. That's why I feel so confident telling you right now that, yes, we are working on this. All this is great and very exciting. I could stop here, but there is one big challenge which still remains. And that challenge is not whether we'll get the data and the data will be good, and maybe we'll even see signatures of oxygen gas. The big problem is that we are not sure that we know exactly what we are looking for. Or let me rephrase that. We are looking for something which resembles Earth life precisely. It produces oxygen gas, the chemistry of the underlying phenomenon, as we call it, life here on Earth, behaves the same way and produces the same signatures. What if life on other planets is not like life on Earth? And so this is the big question, and has been the big question all along, that it is possible that life on other planets is not the same on life on Earth in its basic fundamental chemistry. Even life here on Earth, like this sulfur-eating bacteria, which appear red, are known to produce different gases, like methane in this case. But we are talking about something fundamentally different in this case. So if we don't know it, how do we know the markers for it? And this has always been an obvious question in the search for alien life. And in fact, this was the reason and the motivation why 10 years ago we started the Harvard Origins of Life initiative, which I'm leading. It was to exactly try to prepare for that data and to answer that question. And the people who are still involved with this, it's a small team of about 12 people, this is the leading steering committee of it, is actually trying to accomplish that. And I wanted to tell you here that I'm very happy to be in Spain, and Madrid in particular, because this is where all this started. The Centro de Astrobiología, which is outside of Madrid, started 5 years before us. And we drew a lot of our inspiration from that center here, which is soon going to be 20 years old. In speaking of history, this field has a long history. People have always wondered about what is life and what is the origin of life. But it is only in the last 5 years that for the first time we can talk about a breakthrough in the basic chemistry and understanding of the basic chemistry and the opportunity to answer some of these questions. Is the basic chemistry unique, and hence life like Earth, life will be the same universally anywhere in the galaxy? Or is the chemistry, in fact, robust, but many possible pathways are there? This really happened about 70 years ago with the pioneer work at the time of Stanley Miller, who is pictured here with his equipment. Stanley Miller did something, together with his advisor, Urey, did something very basic and important. He just mixed some gases, which at the time were supposedly the gases which early Earth had, and with water and some source of energy, which was electrical spark at the time, although Stanley Miller then used other forms of energy and the system still worked. He produced some of the basic building blocks, organic molecules for life, particularly amino acids. Amino acids are the building blocks from which proteins are formed, which are basic to the structure of living things. And so that was accomplished 70 years ago, but unfortunately didn't go very far. And very quickly, within maybe 10 to 20 years of that initial experiment, it became clear that there was a very serious problem and challenge with the chemistry. So in order to explain to you what that problem is and how it has been overcome finally 70 years later, I need to tell you what are some of the basic chemical attributes as a system of life as we know it. We call it the minimal protocell model. And this is something which the scientific community agrees upon and has agreed upon ever since the 50s, when the structure of DNA and RNA was discovered. Essentially, any living form on Earth, and we would argue any minimal protocell at the origins of life, should have three basic subsystems. And technically, they're called metabolic, informational, and compartment forming. But in a lay language, the metabolic is simply the structural and functional chemistry that goes on in any living form, in a single cell, for example. And that metabolic subsystem is almost entirely made of a single type of molecules, which we call proteins. Everybody knows about proteins. And the building blocks, the units, the atomic units, so to say, of a protein, they're called amino acids. The same molecules which Stanley Miller produced in his experiments 70 years ago. So that's the metabolic subsystem. It is very important. You can't have a living system without it, but it's produced by, it's based on a very particular singular type of molecules. The building blocks are amino acids. The informational ones, or as we often refer to as genetic one, genetics, informational, is based on molecules that are very specific. They are DNA and RNA. And about 30 years ago, it became clear that RNA itself, which is a cousin of the DNA molecule, can have all the functions which DNA has. So life today on Earth uses DNA, but it is clear from our understanding today that RNA is the primitive initial molecule, and DNA came as a result of evolution later on. So it is enough to say that the informational subsystem could be based on a molecule like RNA. In the building units, the individual units of an RNA molecule are called nucleotides, and specifically ribonucleotides. That's why the R in RNA is for ribo, and it's simply the name of one of the components of those building blocks, which is called ribose sugar, a sugar component. And finally, the compartment-forming subsystem is really what keeps the entire chemistry into a compartment. Compartment, it was shown about 15 years ago, a very important, essentially crucial, to a chemical system being able to evolve, what we call Darwinian evolution. So compartments are considered crucial. They're in the background, those cells that you see there. The building blocks of compartments are called lipids. That's just the name of the molecules which compartment membranes are built from. So what is the problem? The problem that emerged after Stanley Miller's experiments, it became really acute at the end of the 20th century and the beginning of the 21st century, is that while we can make all of these molecules of all the three subsystems, in fact, it's very cheap to buy them on the market today because industrially we can make all of them. We cannot make, didn't know a way to make them all at the same time together. In other words, different and mutually incompatible chemistry are needed to make all of them. In a lab, it's very easy because you make some of them in this part of the lab, another one in another part of the lab, and then put them together. But at the origin of life, on a planet early on without humans and human labs there, this has to happen naturally at the same time. And this is what became known as the chicken and egg problem, and a problem which was really impossible to resolve for 50 years. Not surprisingly, Carl Sagan at the time famously said that we may have the notes, but we still not yet have the music. So what is the approach which turned out to be successful? Initially, after the Stanley Miller experiments, people started saying, oh, we can simply take the simplest bacteria, microbes that we know, and remove some of the DNA genetic information and make them like minimal protocells. This is called the top-down approach. You start with an existing living organism and genetically engineer it to be more primitive. The other approach, obviously, is the opposite. You synthesize the basic molecules, the amino acids, the nucleotides, and the lipids, and try to put them all together and hope that a chemical system behaving like life will develop from this. It made sense originally to try the top-down approach, and people did, and for 50 years didn't succeed very much. The smallest number of genes, which was just published last year, is 473 for this organism, but 1/3 of those 473 genes still have functions which is unknown. So that project essentially is at a dead end after 50 years of trying. The bottom-up approach is the one which our team and other teams have been trying now, which is to build a system which should consist of the basic three subsystems with about 150 genes required for that, give or take. We don't know exactly. And especially if we have the understanding now that DNA is not the primary molecule, and all the functions that DNA does can be accomplished by RNA alone, that makes this bottom-up approach even more realistic from the point of view of biochemistry. And I'll show you just one example of that before I summarize and finish my talk. This one example has to do with RNA and what we call the RNA world. That is the ability to build a minimal protocell based only on RNA as a genetic molecule with very few amino acids and proteins necessary for its functioning. So RNA and DNA are cousins, where essentially they have the same structure with a backbone made of what are called phosphates, phosphorus, with 4 oxygens attached to it. And then there is the sugar, the famous sugar, the blue color, and then the bases. DNA has 2 strands, and they famously form a spiral, while RNA normally only has 1 strand. Although RNA could also form 2 strands and form a spiral. So the big question is, how could you synthesize RNA under normal conditions? And could you do that? In other words, can you make such a string of individual molecules? And I'm not going to scare you with the chemical formulae here anymore. Just let's simplify it with red, blue, and purple for the important 3 components. People, of course, try to do that. And in fact, we know how to do it industrially in the lab. But it was never possible to take the 3 components and simply put them together. The individual components form naturally. We see them even in meteorites. We see them in Stanley Miller's experiments. But if you try to put the three things together, that doesn't work under natural conditions, and even under unnatural conditions. So the big breakthrough, which happened 5-6 years ago, particularly with the work of the Sutherland Lab in England, was that they thought out of the box and they said, maybe we shouldn't try to synthesize the sugar, the base, and the phosphate separately and try to put them together. Maybe you should look for pieces of them coming together. So they started with half of the sugar and half of the base, and when the phosphate is in the liquid, it forms this funny half kind of molecule, which then adds on the other part of the sugar which couldn't be added before. It's still a funny molecule because only half of the base is there, but because the phosphate is in the solution, the other half of the base now can be added. It's still a funny molecule because it has two bonds instead of one, and it should be. But then it turns out if you let this molecule in sunshine, especially sunshine which has UV light in it, then lo and behold, you have the whole nucleotide formed. And this out-of-the-box way of doing it from a chemistry point of view turned out to be so robust that even I can do it. I'm an astronomer and I've tried this in the lab. I can do it. It's very robust. It happens under very natural conditions, which we quite sure were present on the early Earth, even on early Mars. And people have taken it and tried it in many different pathways. And now the whole screen is full of those pathways. And you can't see them, but they produce 12 of the 20 amino acids. They produce 2 of the 4 nucleotides for RNA. And they also produce the lipid precursors. So in other words, finally, we have a possible set of pathways which in natural way, under planetary conditions, without human intervention, can produce the building blocks of all the 3 subsystems, which we believe are required for building a chemical system that behaves like a living cell. And so the lipid bilayers in particular are the ones that form the cell membrane. And my colleague Jack Szostak managed to actually show how this works. The lipid molecules are these strings of carbon atoms with 2 oxygen atoms at one side, which are repelling water. So we call those molecules amphiphilic. They repel water on one side and attracted to water molecules in other words. So when you put them in solution in water, these molecules naturally form micelles. And you know about micelles. This is the reason milk is white, because there's this microscopic little spherules of these lipid molecules which always arrange themselves that way, with the heads pointing outwards and the other ends pointing inwards. So they self-assemble, but there is nothing unusual about this. Salt water does the same thing. And we can't see it through a microscope because it's too small, but this is a simulation of what actually happens. As the concentration of these molecules, of the lipids, increases in the solution, which would naturally occur, they start to bunch, the micelles start to bunch more and more with each other, and at some point form a membrane. What you see here is a projection, so this is more like a membrane like this, which then, as it grows, starts forming a cup like this, like this. Eventually, that cup closes completely into a spherical form, and this is your bubble, like soap bubble, except it's inside water solution. It self-assembles. You don't have to do anything. It does it on its own, just as the concentration increases. The microscopic images look very fuzzy. You can't see the individual molecules. But what Jack has managed to do is to show that these behave like really cells in what we know about life today. They let the small molecules, which we are forming with the other chemistry, in, and then inside they grow and stay always inside the cell because they cannot leave now the boundary. It's a semi-permeable membrane. And so what we have finally is a new chemistry of self-assembly that works under conditions which we see or know on other planets. So on one hand, the astronomers have now the ability and the technology to look for this kind of life on other planets. And the chemists and the biologists in the lab have finally come to the point where they're very close to having a chemical system which works and behaves like life. And so the moment we have the ability to measure the conditions on other planets, we will be able to compare the two and essentially say under what conditions would a system that behaves like life will be able to be synthesized. And are those conditions that we see on planet X or X2 comparable or similar to those conditions? So this is the confluence of two major milestones and breakthrough in individual parts of science, which we are now trying to use together in order to not only do the measurements and search for life on those nearby planets, but also being able to understand those measurements and hopefully have the confidence to say that we have found evidence for life when we do, and if we do. We still don't know what we are going to find. So we still don't know how unusual our solar system and the Earth is in terms of the conditions on the surface. This is what is going to happen first. With those big telescopes, in the next 5 to 10 years, we are going for the first time be able to compare the atmospheres of Earth-like planets around other stars with the atmosphere of the Earth today or in its history. And the other one is with the work which is done in the lab, we hope that we'll be able to see and know we are seeing life when we're actually looking at it, which is not necessarily true right now, before we have done this homework. So thank you very much, and stay tuned for what is going to happen next, which is a very exciting science in the next 5 years. Thank you.